Español? Okay. So I will introduce you in Spanish.
Okay.
So es mi gran placer de introducir a Raphael Mercier, que empezó su maestría en genética de plantas, tuvo su degree en 1998. Hizo su doctorado en biología de plantas en la Universidad Paris-Sud de Saclay y obtuvo su degree en 2001. Después se fue por 2 años en la Universidad de Birmingham a hacer un postdoc en la meiosis de Arabidopsis. Y es PI desde 2003. Después fue director de investigación desde 2010 hasta 2019 en la unidad Institut Jean-Pierre Bourgain de INRA de Versailles, donde continuó su trabajo estudiando la meiosis de Arabidopsis principalmente. Y le voy a mostrar que tuvo varias investigaciones que realmente abren qué conocemos de procesos fundamentales de la meiosis, especialmente sobre los crossing over, los entrecruzamientos. Y vamos a ver, creo, en su presentación que Arabidopsis aún puede ser un modelo que trae información que puede ser generalizado a varios modelos. Y en 2019, Raphael se movió como director del Institute for Plant Breeding in Genetics del Max Planck Institute de Colonia. thank you very much, Raphael, for presenting here, and I hope you will enjoy what Raphael has to say about what limits meiotic crossing overs.
Thank you, Arnaud, for the introduction. Thank you very much for the invitation. I cannot continue in Spanish. I don't speak Spanish at all. I can do it in French, not in German, but I can do it in English, of course. So indeed, the topic of my talk is what limits meiotic crossovers. So I like to start with this picture here, who is, to my knowledge, the family with the largest number of children. So that's the Radford family. They have a single couple at 21 children. So that's quite amazing, first of all, to have 21 children. But what is also amazing that each of them is unique. You have all these different humans being here, who are all different from each other. And why are they different from each other? Because of meiosis. So you are unique. Everything you eat, all your models, if you work with eukaryotes, all individuals are unique because of meiosis. Because during meiosis, you have shuffling of chromosomes and recombination between chromosomes that make each genome of the new individual unique. And if I focus really on meiotic crossovers, this exchange between homologous chromosomes that create new chromosomes for the next generation has two consequences which are crucial in biology. The first one is shuffling of genetic information. So in this very historical example in flies, this combination of phenotype is possible only if you had a crossover right in between those two genes. So showing that recombination enhances genetic and phenotypic diversity. And crossovers have a second function, which is very important too, which is a physical function, a connection between the chromosomes, which is established through this exchange of genetic material. And this is absolutely required in vast majority of eukaryotes for proper distribution of chromosomes during meiosis. So, for example, in human, most aneuploidies at the origin of trisomy, for example, are due to lack of crossovers, mainly in female meiosis. So showing how important is the physical function of crossovers. So, and when we look at crossovers, eukaryote-wide, there are a few features that clearly emerge. One is the so-called obligate crossover. So we observe at least one crossover per chromosome pair at each meiosis. At least one. This is, of course, connected to the physical function of crossovers which are needed for proper meiosis. But we don't see many. Really typical meiosis is 2 to 3 crossovers per chromosome per meiosis. And to convince you of this observation, I have here a plot with many chromosomes from different eukaryotes. So each dot here is a chromosome, each color code is one species, and the X scale here is the physical size of these chromosomes, and the Y scale is the number of crossover per chromosome. So first thing you can see that the very vast majority of chromosomes receive between 1 and 3 crossovers per generation. And this is completely independent of the physical size of those chromosomes. So here, for example, you have button mushroom or trypanosoma, typically 2 crossovers per chromosome per generation. And here, on the other end, you have barley or opossum, and chromosomes are almost 1,000 times bigger, and they also receive only 2 crossovers on average per meiosis. There are exceptions with exceptionally high level of crossovers in Pombé, in Cerevisiae, or in honeybee, for example, but those are really exceptions. Most eukaryotes, including my favorite model, Arabidopsis, and very, very likely your favorite eukaryote model is here with typically 2 crossovers per meiosis per chromosome. And the last property that is observed of crossover at meiosis is their distribution along chromosomes. When you have several crossovers, like here, they tend to be away from each other. They repulse each other. somehow. So this is called crossover interference because the presence of one crossover interferes with the presence of another crossover. And amazingly, what I just said was defined more than 1 century ago by the first geneticists working with Drosophila. And they defined completely crossover interference, identified the two ways to look at it, like the lack of double crossovers close to each other, but also the other way to see that the presence of one crossover inhibits the formation of a second one. And despite this description more than 1 century ago, we know very little about how this interference is implemented. What is the mechanism of crossover interference? I will go quickly into the molecular mechanisms of crossover formation. Just a key word that I'd like you to keep for the rest of the talk. So first, recombination at meiosis initiates by formation of double-strand breaks of DNA, so cut in the DNA, and actually hundreds of them. A very large number of cut of DNA at the beginning of meiosis. And those breaks are repaired catalyzed by those two proteins here, and we use DMC1 as a marker of recombination. You can see here, each little foci dot here is one double-strand break being repaired. So in thaliana, we estimate roughly to have 200 of those breaks being repaired at the beginning of meiosis. Similar numbers are observed in mouse, for example. And from this very large number of double-strand breaks, only a few ones go down that pathway to make crossovers. And actually, there are two pathways that make crossovers. A major one here, called the class 1 pathway, or the ZMN pathway. And among M is a protein called HI10, and I will talk a lot about that protein today. And then those make interfering crossovers. And there's a very minor pathway making only one crossover on average per meiosis. And the vast majority of those breaks being repaired are actually repaired without making a crossover. So a very large excess of double-strand breaks, of precursors compared to the final number of crossovers. So crossover number is very low in most species, despite a very large excess of molecular precursors. So a question I've been addressing in my team for years now is what limits crossover formation? What are the mechanisms preventing more crossovers? And to try to think how this is implemented, I show you like the way we think about crossover designation. So initially on one chromosome, you have plenty of double-strand breaks. And some, by an unknown mechanism, are designated to become a crossover. Some mysterious signal is transmitted along chromosomes, preventing the double-strand breaks to become crossover. And then another one can, at a certain distance, become a crossover, and again, prevents the neighbor to become crossovers. So those are class 1 crossovers. Here we are. So they are not numerous and away from each other, so interfering. But there are some rebels, and among the intermediates which are supposed to become non-crossovers, some become class 2 crossovers. You can see class 2 crossovers as a kind of backup pathway. In case something went wrong, then you can still repair the double-strand breaks, making a crossover. This is less regulated, more like a backup. So back to that question, what limits crossover formation? The first approach we took to do that is to design a screen to identify specifically factors that prevent crossovers. So for geneticists, what you need to make a good screen is to have a good phenotype. And what we used is the fact that mutant in the main pathway of crossovers, so one of them here, have very little number of crossovers. As you can see here, very little connections between chromosomes, because as I said, crossover make connections between chromosomes. And because of this lack of connection, you have random segregation of chromosomes during meiosis, and then strongly reduced fertility. So we use this strongly reduced fertility, which is visible in thaliana by the short fruits, and apply mutagenesis, looking for plants with restored fertility as a proxy for restored crossovers, like being likely a mutant in an anti-crossover function, because the mutant will have more crossovers. So we did that at a large scale and identified altogether 3 pathways, so regrouping a number of genes. And those 3 pathways are, the central proteins are the helicase FANChem, the helicase REQ4, or Bloom in humans, and the protein FIGL1. And in the corresponding mutants, we see an increase of recombination. So I show that here with one of the tools we use to measure recombination, which is very nice, which is based on segregation of colors in pollen grains. So this is based on 3 markers here, 3 transgenes conferring different colors to pollen grains, and depending on how they segregate, you have a different distribution of colors in the pollen grains. And then based on that, we can measure recombination in those intervals. So you can see that we have an increased frequency of recombination in all 3 main mutants I've described, FIGL1, FANChem, and REQ4. And this is indeed 3 pathways, because when we combine 2 by 2, every time we have additional effect of crossover formation in those mutants. And the champion is that one, the FIGL1 REQ4. And then we switch to a genome-wide level, which is like making hybrids between 2 different strains and sequencing the progeny. And by sequencing the progeny, we can detect crossovers and then have an access of crossover distribution and numbers genome-wide. And then we got this kind of crazy map, which is here, we are just along one chromosome 3 in that case, and you see that you have a variation of number of crossovers in wild type here. And you see here the frequency of crossovers in REQ4 FIGL1 double mutant, which is 7-fold increase. So I've showed you that the variation in eukaryotes is quite limited. And here, just with 2 mutations, we increase the number 7-fold, which is really a huge increase. And despite of this huge increase in those mutants, the fertility is just fine. So having more crossovers is not detrimental for meiosis. Next, we looked at where those crossovers are coming from, and I'll just show you one piece of data. We have one marker here, which is specific to class 1 crossovers. And in all these mutants, the number of class 1 crossovers is not modified. And we have other data to I will not show today for sake of time, but that clearly shows that what is increased in, I've missed one slide here. Okay, what is increased in those mutants is specifically the class 2 crossover, which is normally very, very minor. So what we think is happening in wild type, you have those 3 proteins, FIGL1, REQ4, and FANChem. All those 3 push those recombination intermediates towards non-crossovers. So in absence of those proteins, then you have specifically increase of this minor pathway, class 2 crossovers, but without touching the class 1 crossovers. So the model we have for those mutants, so we have everything is normal for class 1 crossovers. They are not numerous, they are away from each other, but all those intermediates which are supposed to become non-crossovers become now crossovers by the class 2 pathway, by the backup pathway. So we can increase crazily crossover number by increasing the normally very, very, very minor pathway class 2. But the big question that we are addressing now is what controls class 1 crossovers? Because those one are correspond to the major pathway in wild type. Those ones are the ones which are interfering, which told to each other to be away from each other. And this is the mystery we are trying to solve here. How this dialogue between crossovers is established. And this distribution of crossovers during meiosis is established within a very specific structure that occurs only during meiosis, which is called the synaptonemal complex. So just to make it simple, this is a protein structure that organizes the chromosomes. So one chromosome is organized in loop in one of these axes here, and the homologous chromosomes organized in loop in the other axis here. And the two axes are zipped, really like a zipper. You can have this image in your mind. You see the zipper that zip the two homologous chromosomes together. And within that very specific structure, crossover are made and controlled. And to make it more real, this is a super resolution microscopy of chromosomes during meiosis in Arabidopsis thaliana. So the two purple lines are the two homologous chromosomes, and the green signal is a protein that zip the two homologous chromosomes together. And because it's super resolution microscopy, we can even zoom more and establish the organization of this structure. So first here, we mark the two axes, the two purple lines, if you wish, here, and there's the two external lines here on this picture. And then we mark the central protein, which is called ZIP, but we mark it with two different antibodies, one at the C-terminal end of the protein and the other antibody at the N-terminal end of the protein. So here, what we see is the two ends of the same protein, which is this ZIP1 protein here, which is organized in coiled and make in dimer, making this transverse element that zips the two homologous chromosomes. You can see here, so you have the N tail of the protein in the middle, the C tail of the two end of the two dimers in the external side, and then the two axis of the two homologous chromosomes. I have to admit that this structure is very canonical. The same has been described in many eukaryotes, including mouse, Drosophila, yeast, etc. Nothing special in thaliana about this organization. This is very conserved in eukaryote. So this was my first player of crossover control, the synaptonemal complex. The second that I've mentioned a bit before is HITEN. HITEN is one of the pro-crossover function, pro-crossover protein, one which is needed to make the class 1 crossovers. Okay. But in addition to that, HITEN has a very interesting behavior during meiosis. So I will show that to you that now. So here, again, I use immunolocalization with super resolution. In purple here, you have the central part of the complex, ZIP1, and on top of it, you have HITEN. And HITEN initially makes multiple foci like this. And with progression of meiosis, it's more and more non-homogeneous, with brighter, larger foci forming, and other regions being progressively having less and less of the HITEN signal. And this continues to end up with some very bright foci along chromosomes and almost nothing left on the rest of the chromosome. And very interestingly, those bright foci of HITEN that we can see here now colocalize with bright foci of MLH1, which are a marker for crossover site. So those big foci marks the site of crossovers. So this behavior and other things inspired a model for crossover interference. And I have to say, this is very fresh. I mean, this is really an idea that emerged in the course of last year. So the idea is that you have progressive coarsening of droplets of HITEN. So initially you have these multiple droplets, and then the molecules coarse into bigger and bigger droplets, or bigger and bigger foci. And I'm working with David Ficker, collaborating with David, with a theoretical physicist. And here I will show you a very, very simple model. So we just distribute some small droplets here along this space. And the only thing you have to implement as a property is, or the two things, is first diffusion of molecules. They can diffuse in this purple space freely. And then the property of droplets, the bigger droplets tend to attract more molecules than the smaller one. This is described in many systems in physics. There's several mechanisms that can lead to that, but it's not important what is the mechanism today, just the property is important. Bigger droplets tend to obtain more molecules and smaller droplets. And just implement this, and this is what is happening. You will see that with time, smaller droplets will tend to disappear. You have competition between droplets of different size to attract molecules, to capture molecules. And with time, we will see that a certain pattern will be established with a very small number of big foci first. And then those big foci tend to be away from each other because they used up the material next to them. So this predict very simply large foci which are away from each other, which are interfering, if you wish. So this is the model we are testing now here. So heightened foci for multiple droplets and coarse with this diffusion mechanism of heightened constrained by the SC, and then leading to this patterning of future crossover sites. Okay, one additional features about the synaptomal complex, which I show here to you here again, is that in Arabidopsis, it is different in male and females. It is shorter in female than males. And this is kind of intriguing because Arabidopsis is hermaphrodite. This is the exact same individual that we look at here in male and females. So the genome is the same by definition. This is the same individual. But the organization of the complex is different, making the chromosomes longer in male than females. And quite intriguingly, this is correlated with the number of crossovers. We have also more crossovers in male than females. So this is something which is relatively common, to have a different number of crossovers between male and female of the same species. In thaliana, this is more in male than females. In human, this is the opposite. There are more crossovers in female meiosis than during male meiosis. And we see the same correlation also. This is here based on foci of MAH1 that I've told you is a marker of crossover sites, but we see the same thing with the same proportion. If we do by genetics, by sequencing progenies, we see also more crossovers in males and females in the same proportion. Another difference when we look at crossovers between male and females is the strength of crossover interference. So I've introduced a bit before that crossover interference is the tendency of one crossover to prevent another crossover to form nearby. So we need to have some tools to measure that. And this is my favorite way to measure crossover interference. So what you do, you take two intervals, you look at crossover frequency in both of them, and then you can predict how many double crossovers you will expect if they will be independent. And you can compare that to the actual observation of double crossovers. So this is called the coefficient of coincidence. If double crossover will occur as expected by chance, this line will be flat at 1 because we make the ratio between the observed versus the expected. But what we see in real life is this curve here. Like close intervals tend to have very, very few double crossovers, much less than expected by chance. But when we consider intervals which are away from each other, then we start to see again double crossovers. And you can see that in female, in pink, the crossover interference affects a larger part of the chromosome than in male. It takes more distance to get back to close to 1. The crossover interference is stronger. And all those properties I've just described are very well predicted by what we call now the coarsening model. Because longer SC, longer chromosomes, is predicted to have more foci than shorter SCs, shorter synaptomal complex by this system. And also crossover interference by modelization is predicted to affect longer distances on shorter axes, observed in females, than in males. So it fits pretty well to explain the male-female difference. Okay, let's continue to challenge this model and to see if it resists to modification of the parameters. So the first one we test here is to overexpress the heightened protein. If we overexpress the heightened protein, we will have more molecules loaded along the centromeral complex, both in male and females. So this predicts a higher number of crossovers. But it also predicts to keep a difference between male and females. This should proportionally increase crossover numbers in male and females. So we tested that using a line developed in the lab of Jan Andersson a few years ago. And indeed, in this line, we observe more crossovers, more in the mutant or in the overexpressing line than in the wild type. As you can see here, you have male females of wild type and you have male females of the heightened overexpressor line. You have a clear increase, female to female, male to male, but you can also see that we keep the male-female difference, exactly as predicted. This was just foci. We also confirmed that again by genetics, by sequencing progeny to detect crossovers. We see an increase in the heightened overexpressor compared to wild type. And again, we keep the difference male-females. So what we see in cytology with foci is completely confirmed by genetics, by looking at genetic crossovers in the progeny. What about crossover interference? Here we have crossover interference in wild type, and here we have crossover interference in the heightened overexpressor. So you see that crossover interference is less effective, is reduced in the heightened overexpressor. You see, now you reach back to 1 at shorter distances compared to wild type. But still, you keep a difference male versus females. Females is more affected, has more crossover interference than males. And again, this is exactly what is predicted by the coarsening model. So I won't go into the detail of these plots here, but using the same parameters, we can fit perfectly well the observation of number of foci per cells, the number of crossover per chromosomes, and crossover interference. So this way of modeling coarsening predicts pretty well the behavior of crossovers in male, female, of wild type, and heightened overexpressor lines. But next, we can test something more. What will happen if we get rid completely of the structure that control crossover distribution? If we make a mutant of the SC of this structure, a mutant of the Z1 protein I've introduced, the one that makes the transverse filament. So this is the Z1 protein here, the transverse filament. There was a little complication that prevent us to do that for some years or in the past, that the protein coding this is a recent tandem duplication. So in thaliana, with 2 genes, which are both functional, both perfectly redundant. It was very difficult in the pre-CRISPR area to make those double mutants. Actually, we tried, never managed to. But with CRISPR, then we were able to make double mutants and even full deletion of the locus to be sure we have null mutants. What do we see in this mutant of ZIP1? So ZIP1 is a protein that we mark here in the wild type. We have previous slides in red and green here. Now, in the mutant where this gene is mutated, of course, we don't see that signal anymore, but also we see the 2 axes, which are now at a quite large distance from each other. They are still aligned, they still talk to each other, but at a longer distance. They are not anymore zipped by the ZIP1 protein. What happened to crossovers? Do they occur? Yes, they do, but they occur in higher numbers. On the ZIP1 mutant, when this structure is absent, not only we see crossovers, but we see more crossovers. So this is what I show here. You see, again, this foci that mark crossovers. I remind you that in the heightened overexpressor, we had increased compared to wild type, but keeping a difference between male and females. Now I show you the ZIP1 mutant in blue, and you see we have an increase compared to wild type. But the key difference is now we have the same number of crossovers in male and females. So the difference is gone. We confirmed again by progeny sequencing. So wild type, we have male-female difference. High-10 overexpressor, we have male-female difference. In the Z1 mutant, now we have the same number of crossovers in male and females. So this is the model here in the Z1 mutant. So in the wild type and high-10 overexpressor, you load high-10 along SC. The high-10 diffusion is constrained by the C-terminal complex. Okay, I tend to say SC all the time. SC is the C-terminal complex. So high-10 is diffused along the C-terminal complex and coarsened into small foci, into large foci that determine the crossover sites. But in the absence of ZIP1, in the absence of the C-terminal complex, now the diffusion of high-10 is free in the nucleoplasm. And then it still makes big foci, but the system is not completely blind to chromosomes, because what measures the chromosomes is the C-terminal complex in this system. So now you can have crossovers, but they can be at any distance from each other because the system doesn't see anymore the chromosomes. Okay, so we modify the system by two ways so far, by increasing high-10 levels, which increase crossovers, by making mutant in ZIP1 that also increase crossovers, slightly different manner, but both increased. So what will happen, and we see that both in pure line Colombia or in hybrids, and what will happen now if we combine the two together? So in the ZIP1 mutant, no central complex and high-10 overexpressor. And here came even a surprise also for me. We had a massive increase of class 1 crossovers numbers, really huge increase I can see here, in both in the pure line and in the hybrid. So you may say these are just foci, maybe they are not true crossovers. So again, we sequence progeny and we still see, or we do see, you confirm this massive increase of crossovers compared to wild type. I can show you here, this is female gametes. We have an average of 20 crossovers in a single gamete compared to 3 on average in wild type. So to try to share with you how I'm surprised by this result. This is what you see when you sequence or analyze crossovers in a gamete, in basically any eukaryote. You see 1 or 0 or 1 crossover per chromatid here in the gamete. And look what you see in those mutants here now. You see this chromosome just crumbled with many exchanges, which are the crossover sites. That's really a complete deregulation of the class 1 crossovers, which are normally heavily regulated. And again, fertility is normal. So we can increase also the class 1 crossovers without visibly affecting fertility. So the take-home messages of the different parts of my talk. First, class 2 crossovers, which are kind of backup pathway or DNA repair pathway, very similar to somatic pathway of DNA repair, are actively prevented at meiosis by 3 mechanisms, FANCM, REQ4, FIGL1 being the key actor of that. So the crossovers which are normally, those crossovers are actively depleted during meiosis. The class 1 crossovers, the one which are important for meiosis in wild type, are regulated, we propose, by 2 main parameters. The C-terminal complex, in particular the length of the C-terminal complex, and the dosage of high-10. And these 2 parameters are really well predicted in an emerging model of crossover patterning and crossover interference, which is we call the coarsening model, that explains very well crossover patterning in both wild type and mutants. And this is maybe what I've shown so far is very consistent with this model. we cannot completely exclude completely other models now, but from my perspective, this is the most tempting model I've ever seen on crossover interference. And I can say it fairly freely because I'm not the one who developed it initially. And this is maybe the solution to a century mystery. That's why I'm so excited. And finally, we can show that crossover can be massively increased without immediate dramatic effect. So it raises the big question is, why having so few crossovers in almost all eukaryotes if it's so easy to increase their numbers and there is no effect on the immediate effect when we increase those crossovers numbers? So it's quite intriguing, and if you wish, we can maybe speculate on that in the discussion. And with this, I'd like to thank the people who have done the work. So mostly what I've shown today was mostly done by Stephanie, who has done most of the cytology of SIN. Laia was produced the ZIP1 mutant and analyzed the ZIP1 mutant. Chichao, with an extremely talented bioinformatician who has analyzed all the crossover data. And Julie, who has developed the method to trace the chromosomes in 3D. And I'd like to really thank this team, which is very dynamic. And we are always looking for people to join us. Don't hesitate to contact me if you want to join us. And with this, I'd like to thank you for attention, and I'm ready to take any questions.
Thank you so much, Raphael. Any questions? I will start with one, probably, since we are looking for more. So how do you, well, how do you see ZIP1 with the effect of
Heighten.
Do you see like a roadway in order that it cannot move away from this? Or what kind of physical parameter do you expect from this interaction?
That's okay. That's interesting. So that's mainly speculation I will do now. But indeed, to this model to work, we need heightened diffusion to be constrained within the centrosomal complex, and ZIP1 is required for this constraint. Initially, we see the foci really in the middle of the structure. So either that's a direct interaction between the end terminal of the ZIP1 and heighten, a weak interaction enough to constrain the protein here, but not to make it fixed. Or, and maybe I'm more in favor of that, several proteins have been described being associated with the center of the complex in many eukaryotes. So my wild speculation would be that those proteins create attraction zone for heighten, and heighten it there, but can still move along this structure. So it has to be protein. I will suspect protein interactions, but not very strong protein interactions to support this diffusion, this constrained diffusion.
We have a question from Joseph Dubrovsky. Joseph?
Yes, hello. Just very, very interesting talk. Thank you very much. I want to ask you, what is known about mitotic recombination? Are these factors participate also in mitotic recombination?
So the first three factors I've described for the so-called class 2 pathway are also involved in somatic recombination. So they are involved in DNA repair, because double-strand breaks can also occur during somatic life for different reasons. And this is really a repair pathway that typically tend to not make crossovers, but sometimes make crossovers. And really those, what we call class 2 pathway in meiosis is actually a somatic pathway, just a repair pathway. But all those other factors, the class 1 pathway with ZIP1, heighten, and many other factors I haven't described today, are specific to meiosis. So what is really specific to meiosis is this class 1 pathway, this heighten pathway, that as a new property of controlling the number and distribution of those crossovers.
All right. Mm-hmm. Thank you very much.
You're welcome.
So there is a question in the chat. Do you see it, or should I read it?
Yes, one second, because the second question has pushed the first one, it disappeared. Okay. Why are there more crossovers in male and female in thaliana? Is interference expected to be higher when the AC is larger due to AC interference? So, yes, so the prediction here, we have a shorter axis, but a shorter cylindrical complex in females than males. And the model predicts that the distance in micrometer of complex in male and females, which interference acts, is the same. So let's say you act at 10 micrometers, it will be the same in male and females. But because the chromosomes are more compacted in females, then this will affect a longer distance in genomic space. And that's why crossover interference appears stronger in female compared to males. And that's why you have also less crossovers. So all is based on the fact that this diffusion space is shorter in females than males. And this correlation between length of the AC and crossover number is conserved in several eukaryotes, including humans, when it is the opposite. Longer chromosomes in female and longer and weaker interference. Okay, the next question is very interesting, that I would love to have a possibility to have the data to address that. So the question is, how do high-10 and ZIP1 mutants behave under reduced DSB repair conditions or under reduced DSB formation? Okay, this is one for the less aficionados of meiosis. There is one mechanism called crossover homeostasis, which is, as told you, there is quite an excess of double-strand breaks compared to crossovers, and this can be reduced or increase the number of double-strand breaks without affecting very little the number of crossovers. This is what is called homeostasis, because it is buffered. You can modify the number of double-strand breaks, and you have stable number of crossovers. In ZIP1 mutant in particular, I suspect this is affected. I will say that because in the ZIP1 mutant already, we see some chromosomes without crossovers, because you lose the control of between chromosomes, because the system is blind to chromosomes. So if we reduce the number of double-strand breaks, I suspect we'll also reduce the number of crossovers. But I don't have a good mean at the moment to have reduced double-strand breaks in Arabidopsis. We have only null mutants, to my knowledge, for double-strand break formation. Okay, one more question. What is the molecular mechanism of SC-mediated interference? Is DSB formation inhibited, or are those that do form and produce? Okay. Is DSB formation or are those? Okay, so I explain one more time the model, which is you have many double-strand breaks, and then along the chromosomes, and then on top of that you have coarsening of high-10. And indeed, we need to have some way to ensure that every time we have a high-10 foci, there is a DSB or DSB repair event under that foci. That probably we can also speculate that the proteins involved in this DSB repair can also interact with high-10, and we know some of them already that are good candidates for that. Then we'll have one foci of high-10 surrounding an intermediate of recombination, and this intermediate will be protected by high-10 and partner to make a crossover. And every intermediate which is not protected by high-10 and partners will be then subjected to non-crossover pathways and will be cleaned without making crossovers. That's the idea. So the winner inside and the loser outside of the droplets, if you wish.
Questions? Okay. So from YouTube, you have a question from Juan Enrique Moret. Thank you for your talk. It seems anti-intuitive to naturally have few crossovers, since they could contribute to segregate deleterious mutation easier, especially considering how easy it is to increase the number of crossover. Could it be the low crossover is beneficial from an evolutionary point of view?
That's a key question. Thank you for asking it. Honestly, I can only speculate. So indeed, we observe only very few crossovers in most eukaryotes. So there are a few families of hypotheses. One of them was that having many crossovers will disturb meiosis, will disturb chromosome segregation, and that's why this is counter-selected. Our results and others suggest really that is not a correct hypothesis. So it must be the number of crossover itself which is under selection. The genetic consequence of crossovers with under selection. So crossovers are beneficial to purge deleterious mutation, also to make new combinations of alleles, to have new phenotypes, to explore new niche. So all of this seems to suggest that we should have plenty of crossovers. But on the other side, you can also think that crossovers could be bad if you have accumulated beneficial combination of alleles in cis. And then breaking those combinations is actually a bad idea because you have accumulated with time during generations adaptation to your environment. So you don't want to break those beneficial combinations. So then it suggests there is a balance between create new combinations with many crossovers to adapt, but not to break the previously accumulated beneficial combinations. And then the two together seems to lead to an equilibrium which is very close to a single or a few crossover per chromosomes. But this is quite difficult to modelize. I'm discussing with evolutionary biologists. It's difficult because this is heavily dependent on parameters you use. But that will be at least my favorite hypothesis, a balance of pros and cons of genetic recombination itself.
So the heterochiasmy is doing that to have the two sides of a bit trying to be revolutionary when you make more crossing over and try to on the other side.
Yeah, the heterochiasmy is quite intriguing indeed, because heterochiasmy could be if heterochiasmy will be adaptative, I would expect, maybe naively, but I would expect that it should be the same in all eukaryotes. Like if the optimum for recombination is different for male and females, that we can understand because the reproductive strategy is different for male and females, with very few gametes, with a lot of energy, or many gametes in which we invest not that much. So we could imagine that a different level of recombination will be adapted. But then I will expect the heterochiasmy to be the same in plants and humans. There's no reason I can think of which will lead to that. So actually, maybe at the end, it's maybe just noise, because you have the two, some freedom in crossover levels, and by chance, you can be different in male and females in one direction or another, or to be equal, also by chance, maybe.
So I don't see more questions. Thank you very much for this fantastic talk, Raphael.
Thank you very much for this discussion.
To see you at what point to give a potential seminar.
Looking forward. Thank you.